Hello everyone, welcome to Nolly Learning Center. Today we are going to solve a biology paper 4 of Feb March 2025. Let us begin with the first question. The question says, Figure 1.1 shows the movement of particles through an epithelial cell in the small intestine. In figure 1.1, arrow P represents the diffusion of oxygen molecules. Describe what is meant by the term diffusion. So we can define diffusion as the movement of particles from an area of high concentration to an area of low concentration down the concentration gradient. So this is what we meant by diffusion. Let us move on to the next question. State the type of energy needed for diffusion. So as we all know that when the particles move, they gain kinetic energy. So the type of energy needed for diffusion would be kinetic energy. Next question. Carbon dioxide molecules also move by diffusion. State the name of the process in human cells that produce carbon dioxide. Now we know the process in human cells that produces carbon dioxide is aerobic respiration. So the answer for this will be aerobic respiration. Next question. On figure 1.1, draw an arrow to show the direction of diffusion of carbon dioxide molecules. So we know that carbon dioxide will be greater in the cell and lesser in outer cellular matrix. So in that case, the diffusion of carbon dioxide will be outwards. Next question. In figure 1.1, arrow Q represents another type of particle movement. Identify the type of particle movement represented by arrow Q. Explain your answer. So we can see that Q are the particles moving from a low concentration to high concentration and using energy. So in this case we can say that the type of movement shown by Q is active transport. And the explanation for that will be, as the particles are moving from lower concentration to higher concentration using energy from respiration. Let us move on to the next question. In figure 1.1, particle R moves from the lumen of the small intestine into the epithelial cell. Suggest why particle R cannot be starch. So we can say, let us see, particle R is moving into the epithelial cells. Now particle R cannot be starch because starch molecules are large molecules. Starch is a polysaccharide. So in that case the answer for this will be starch are very large molecules and cannot pass through the cell membrane. and hence starch cannot pass from the small intestine into the epithelial cell. Let us move on to the next part of the question. Figure 1.2 is a photomicrograph of red onion cells. Figure 1.3 is a photomicrograph of the same red onion cells after being immersed in a salt solution. So we can see that the cells in figure 1.2 are intact, whereas when I see the cells in figure 1.3, they have shrunk in size. Let us see the question. Using figure 1.2 and 1.3, describe and explain the difference in the appearance of the cells before and after immersion in the salt solution. So we have to describe as well as explain. So in describing, we'll just describe what is the difference that we can see in the two diagrams, and then we'll explain the reason for the same. So first, let us explain the appearance. So we can say that in figure 1.2, the onion cells are turgid and pushing against the cell wall and are intact in shape, whereas in figure 1.3, the onion cells have shrunk as the cytoplasm pulls away from the cell wall. Which is called as a plasmolysed cell. Now we'll explain the reason for that. The reason for the same can be that the salt solution in which the cells are placed is hypertonic, and hence the cells lose water through osmosis from a high water potential to a low water potential through a semi-permeable membrane. So that's the answer for this 6-mark question. Let us move on to question number 2. The question says the internal body temperature of a person was recorded. Figure 2.1 shows the results. So we can see a graph where the internal body temperature of a person is plotted on the y-axis with respect to the time. Let us read the first question. It says, using the information in figure 2.1, calculate the temperature range for the internal body temperature of this person. So we need to find the temperature range. So we can see that the maximum temperature goes to 38 whereas the minimum temperature goes to 36. So the temperature range for this person will be 2 degrees Celsius. Let us move on to the next question. On figure 2.1, draw a line to show the set point for the internal body temperature of the person. So we know that the set point for the body temperature for humans is 37 degrees Celsius. So we will draw a line here. Next part. The maintenance of internal body temperature is an example of homeostatic control. State the name of the mechanism for homeostatic control. So we know that the homeostasis is controlled by negative feedback mechanism. So the answer for this will be negative feedback mechanism. Let us move on to the next question. It says on figure 2.1, region F shows a change in body temperature. Explain how the body causes the change in the body temperature shown. So let us see in the diagram. We can see that in this change the body temperature is increasing from 36 to 38. So now when the body temperature is increasing from 36 to 38, let us understand what brings about this change. So for this we can say that when the body temperature falls to 36 degrees Celsius, the temperature receptors, which are also called as the thermoreceptors in the skin, detects the change and sends an impulse to the brain. The brain responds to it by sending impulses by the motor neuron to bring about the following changes. The hair particle stand erect, vasoconstriction occurs, and the metabolism increases. Let us move on to the next part of the question. It says figure 2.2 shows a cross section of human skin and the parts are labeled. Let us first name these parts. So we can see that A is the sweat gland, which produces the sweat. B is the adipose tissue under the skin, which comprises of fats. C is the hair erector muscle. D is the thermoreceptor. and E is the air particle. Now let us move on to the questions. State the letter of structure shown in figure 2.2 that produces sweat. So as we marked above, sweat gland produces sweat. So for that the answer will be A. State the names of the structure C and D, which we have already named there. C is the hair erector muscle and D is the thermoreceptor. Let us begin with question 3. The question says, figure 3.1 shows the percentage of water in different structures of the human body. The mean mass of human eye is 28 grams. Using the information from 3.1, calculate the mass of water in a human eye. Give your answer to two significant figures. So let us go for it. So the total mass of the human eye is 28 grams and out of that 95% is water. So to find the answer we can do 95% of 28, which will be 95 by 100 into 28, and when you calculate this value the answer comes out to be 26.6. But as we need to give the answer in two significant figures, the answer for this will be 27 grams. Let us move on to the next part of the question. Describe the importance of water in the human body. So let us write the importance of the water in the body. Very first is, it is a solvent that solubilizes substances in the body. Secondly, blood is majorly water that helps in transport of substances all over the body. And the third point we can say is water passes through the kidneys to remove waste toxic substances from the body. The next question says, cholera is a disease caused by a pathogen in contaminated water. Figure 3.2 is the diagram of the cholera pathogen. Now cholera is caused by a bacteria. So basically this is a structure of a bacteria. Further, state the name of the cell structures L and M shown in the figure 3.2. So we can say that L is the cell membrane as it is the inner layer, whereas M will be the ribosome. Next question. Identify two features shown in figure 3.2 that are typical of prokaryotes. So we can see a flagella present as well as we can see presence of plasmid. So these two are the structures which are typical of prokaryotes. Next, state the type of pathogen that causes cholera. So as we already know that cholera is caused by a bacteria. So the pathogen will be a bacteria. The scientific name for the pathogen that causes cholera is Vibrio cholerae. State the genus name for this pathogen. As we know that in naming, the first name is always the genus name. So in this case, the genus name for the pathogen is Vibrio. Let us move on to the next question. Explain how the cholera pathogen causes dehydration of the human body. So we know when the cholera pathogen enters into the small intestine, so we can say cholera pathogen releases toxins into the intestine that results in secretion of Cl minus ions in the intestine, which results in drop of water potential in the intestine, resulting in movement of water from blood capillaries into the intestine by osmosis, hence causing diarrhea, which causes dehydration. Next question. Complete the sentence about the use of plasmids in genetic modification. So we need to complete the blanks. During genetic modification, human DNA and plasmid DNA are cut with a restriction enzyme. This creates sticky ends, which are joined together using an enzyme called DNA ligase, which modified plasmid containing the human gene is called as the recombinant plasmid. Now let us move on to question 4. Figure 4.1 shows a part of a human placenta and umbilical cord. The arrow shows the direction of the blood flow. The question says, describe the functions of placenta and the umbilical cord shown in figure 4.1. So first, let us write down the functions of placenta. So first of all, placenta acts as a barrier between mother's blood and baby's blood, preventing it from mixing. And allows diffusion of substances from mother to fetus and vice versa. Whereas for umbilical cord, it transports oxygen and nutrients to the fetus and connects the embryo blood supply to the placenta. The next question says, A fetus develops inside an amniotic sac. Describe the functions of the amniotic sac and the amniotic fluid. So first let us describe the amniotic sac. So it is basically a thin transparent bag in which the fetus develops and contains amniotic fluid. Whereas when I talk about amniotic fluid, it is the liquid surrounding the fetus and protects the fetus from mechanical shocks. Next question. Syphilis is a sexually transmitted infection that can be passed on from a mother to a fetus. State the name of another STI that can be passed on from the mother to the fetus. So in this I can say HIV, which is human immunodeficiency virus. Next, state 2 ways to control the spread of STI. So that can be through no sharing of needles and secondly it can be safe blood transfusions. Let's move to question 5. It says figure 5.1 shows a cross section of a leaf. On figure 5.1, draw a circle around one vascular bundle. So we can see one vascular bundle is present here, which is basically the xylem and the phloem. Next, on figure 5.1, draw a label line and letter X to identify the palisade mesophyll tissue. Now we know that the palisade mesophyll is present below the upper epidermis and are cuboidal cells tightly packed, which I can see is present here. Next, state the name of the cell structure in palisade mesophyll cells where photosynthesis occurs. So the cell structure where photosynthesis occurs is called as the chloroplast. Next, describe the functions of the tissues in the vascular bundle of the leaf. So first of all, when I talk about the vascular bundle, it comprises of xylem and the phloem. So let us write about the xylem and the phloem. So first of all, when I talk about the xylem, it conducts water and minerals. Secondly, it supports the plant and is unidirectional. Whereas when I talk about the phloem, it conducts sucrose and amino acids and is bidirectional. The next question says, the leaf shown in figure 5.1 is from an aquatic plant adapted to live in the water. The leaves float on the surface of the water. State the term used to describe the plants that are adapted to live in water. So we know that plants that are adapted to live in water are called as hydrophytes. Identify feature T shown in figure 5.1 and explain how this feature adapts the leaf to float on the surface of the water. So let us see the feature T. We can see that feature T is basically the spongy mesophyll, which is large air spaces, which is very much required in an hydrophyte. So feature T is a spongy mesophyll cell and explanation is it is big air spaces that helps the plant to float. Next question. Explain one other adaptation of these group of aquatic plants. So we can say that the stomata is present on the upper side of the leaf. Usually the stomata is present on the lower side of the leaf, but for hydrophytes the stomata is present on the upper side of the leaf. Next question, that is question 6. Microplastics are pieces of plastic with a diameter that is less than 0.5 centimeter. Figure 6.1 shows the mass of microplastics in the ocean between 2000 and 2040. The data between 2000 and 2020 is an estimate. The data after 2020 is a prediction. So we can see that the amount of microplastic keeps on increasing from 2000 to 2040. So the question says, suggest why the mass of microplastic between 2000 and 2020 shown in figure 6.1 is an estimate. So it is an estimate because microplastics are very small and secondly it is difficult to keep a track or exact count of the microplastic. It is impossible to keep an exact track of the microplastics. Next question. Using figure 6.1, calculate the predicted percentage increase in mass of microplastic in oceans between 2000 and 2040. So we'll take the value at 2000 and we'll take the value at 2040. So at 2000 we can see that the value is around 0.2 and at 2040 the value is around 1.7. So the percentage increase will be 1.7 minus 0.2 upon 0.2 into 100, which gives the answer as 750%. Let us move on to the next question. Phytoplankton are producers found in the ocean. Phytoplanktons absorb microplastic in their cells. Describe what is meant by the term producers. So we can say producers are the organisms which can produce their own food. Next, figure 6.2 shows a shearwater bird. Shearwater bird feeds on fish in the ocean. Suggest how microplastic can end up in the consumer such as shearwater birds. So we can say that shearwater birds feed on fish which may have ingested microplastics, which will build up in the body of the shearwater bird called as bioaccumulation. The population of shearwater birds is decreasing. Describe 3 ways in which population of shearwater birds can be conserved. So first could be protecting the species. Second can be captive breeding programs. And the third method can be monitoring the numbers. Explain the risk to a population if its population size decreases. So what can be the risk to a population if the population size increases? It could be a reduction in genetic variation, can even cause extinction, and lastly, unable to find mates. I hope you all understood this paper. Thank you.